speaker, Professor Koel from IISER, Kolkata. Professor Dash works at the exciting intersection of mathematics, computational neuroscience, and machine learning with research interests that include visual perception, feature extraction, and brain-computer sciences. Her work helped us understand how the brain processes information and how computational models can mirror or augment that process. She has earned several notable distinctions, including the Ramalingoswamy Fellowship from the Department of Biotechnology, India, and Emirates Cal IT Square Fellowship from UC Irvine, and Phi Beta Kappa International Student Scholarship, and earlier academic merit recognitions such as the Engineering Merit Scholarship from Guwahati University and the National Merit Scholarship from the West Bengal Board of Secondary Education. Her talk today is titled Understanding Cognitive Neuroscience Through the Lens of Machine Learning. Please join me in warmly welcoming Professor Koel.
Let me start by thanking the organizers and Abhishek for inviting me to give a talk. So I'll just start off with a disclaimer that I'm not a mathematician by training. In fact, I'm an electrical engineer. In my PhD, I started working with a biomedical engineer, and my work was mainly with signal processing and machine learning using brain data. And then I went on to do my postdoctoral studies in psychology and brain sciences, where I actually learned to do experiments. And then now I am faculty at the Department of Mathematics and Statistics at IISER, Kolkata. But my work is very interdisciplinary. And I could, I guess, pass off as an applied statistician if need be. But I work mainly in the area of cognitive neuroscience and machine learning and computational neuroscience. So we try to sort of make sense of trying to understand the underlying brain mechanisms behind some cognitive processes. So in this talk, what I'm going to do is start off with a very basic introduction about cognitive neuroscience in general, and then see how we can apply machine learning in cognitive neuroscience, and then I'll do a very basic, classical machine learning approach, so that because nowadays machine learning sort of has been translated to deep learning, so as a result, many of the classical methods are ignored. So I thought I'll just give a very brief intro to some very classical machine learning methods, and then see some small applications. Okay. Alright, so let us start with the definition. So unlike maths, where the definition is really rigorous, and that is probably the first question whenever we are interviewing any candidates, so define something. But in cognitive neuroscience, the definition, the answer to the definition actually depends on the background of the person who is answering. Okay, so, but I'll sort of start to make it a bit more generic. So cognitive neuroscience is the sum of two fields, cognition and neuroscience. So by cognition, what we typically mean is application of knowledge, storage and adoption of knowledge, and then essentially mental processes. But intuitively, you can think of it as how we feel, how we remember, how we learn, things like that. Okay. So neuroscience typically means a scientific and systematic way of studying the nervous system. All right. So in the classically there was a there is neurobiology and neuroscience. So neurobiology typically referred to looking at the problem at very basic level of cellular and molecular level. So there was like hardcore biologists working, and neuroscience typically involved looking at it at from a distance, from a tissue level or from a level of the organisms. But nowadays both these terms are sort of used interchangeably. Intuitively, you can think of it as how brain works, right? And then cognitive neuroscience essentially is a scientific and systematic way of looking at this mental processes, the mechanisms behind it. And you can think of it as intuitively as looking at the brain to understand how our mind works. Okay? So actually the history of neuroscience is started somewhere, 6,500 BC, where the first skulls were found with perfect gold. Okay, so that was believed to be one of the first instances of neurosurgery, and it is called trepanning. All right, but the most well-known documented part of neuroscience came from a surgical papyrus, which was believed to be around 1700 BC. It was discovered by a famous Egyptologist called Edwin Smith, and it is known as Smith's papyrus roll. So it was all written in hieroglyphics. Okay, and this is the first time there is a written documentation of the word brain. So this is brain in hieroglyphics. Okay. And the author is supposed to be this demigod and high priest Imhotep, and for those here, it is the same Imhotep of the Marvel franchise. This is the old version, the new one is different. Okay, so this papyrus roll described a very detailed series of 48 case studies where all neural surgeries were performed. And unfortunately, as you know, Egyptians used to store the important organs, but they did not store the brain. So there were two types of schools around this time. One, they thought that brain was important, and that was the seat of intelligence or cognition. The other philosophy was that brain had nothing to do with cognition, it was the heart. Okay, so the Egyptians sort of believed that heart was the seat of intelligence, and hence they did not store the brain. Okay. So Aristotle also believed in heart being the seat of intelligence. So before that, this Greek school of scientists, philosophers thought that brain was where all the mental processes, mind and brain are linked. But Aristotle sort of differed from his teacher Plato, and he said no, brain, the role of brain is just like a coolant. It is there to cool the body. Heart is what is important, and because again Aristotle being Aristotle, he had some definite sort of ideas of why this is so. Because he said that heart is the first organ that is formed when we are born, and that is the last organ to go off when we die. Okay, even if our brain doesn't work, we don't die. Right? We can be in coma for ages. So he gave certain reasons why he thought that heart was more important. And we also have this in our own civilizations of, say, learning by heart. So these were also common. Okay, so this was a thought that mental processes are different from brain. Okay, and then there was this other debate in neuroscience that whether the brain functions as a whole together or it has modules, and each module has some function of its own. So, if the people who believe that brain functions locally, okay, they were called localists, and the other group believed that it functions as a whole. But in the around 1870, I believe, or so, very similar time, two cases were discovered. So one stroke patient came to Broca, so he had an injury in his brain. And he could understand everything, but he could only say one word, that is 10. Okay, so this Broca's patient had an impairment here. So here this part of the brain was not working. At the same time, there was another stroke patient who came to Wernicke, and he had the other problem. He could speak anything. He would just speak nonsense, but he would not understand it. Okay, so that was called the Wernicke's area. So that sort of put this holistic theory to dust, because if the brain acted as a whole, then such a thing will not occur. Okay, so these two patients who appear very similar times sort of, then made way for the local theory. But currently, what we know is that mind and matter indeed is related, it's nothing to do with heart. And instead of localist versus holistic theory, what we know is that brain works in modules, but these modules talk to each other, so they are connected. Okay. So currently what we have is called brain-computer interface, which I'll talk about, and cyborgs. So let me just give one sort of anecdote. So there is this professor, I think he's the director now of some university. So he was the first cyborg. So he, in 1998, implanted a chip in his arm, with which he could control like very basic things like opening the doors and things like that. And then in 2000, he went further and inserted a 100 electrode array in his hand. And later on, he also had a smaller array implanted in his wife's hand, so that both of them could communicate. Okay, so he claims to be the first cyborg. Okay. But coming to brain-computer interface, so brain-computer interface, the idea was that it is to develop a protocol or an interface which does not involve our muscles. So it's a non-muscular pathway with which the brain can interact with the outside environment. So the idea was that there were many patients who their mental ability is perfect, but due to some accident, some spinal cord injury, they couldn't walk, or they're paraplegic, or they're paralyzed. But if we could decode their intention and then somehow connect it to external world, maybe with a robotic arm or a computer, then we can help these patients to communicate with the world. So you can think of Stephen Hawking. So that's a prime example. Okay? So that is the idea. So it sort of started around late 1998, 90s and 2000. That is where these ideas sort of developed. Okay? And although we started there, but Hollywood actually started long back. This idea of thought-based control. So how many of you have actually heard of this movie called Firefox? Anyone? Oh, wow, I'm impressed. Normally no one knows about it. Okay, so this is a Clint Eastwood movie where it was during the Cold War era, where there was this fighter jet which the US wanted to steal from the Russians, and the fighter jet could be controlled by thought. But the trick was that you had to think in Russian. So then our hero, of course, had a Russian mother, and he could sort of get this here. But coming to more recent, or not so recent, so if you think of the Matrix trilogies, there also you see that you enter a simulated world by connecting something similar to electrons. Right? So moving on from fiction to reality. So 2003, a lab at Duke University in North Carolina in USA, they actually showed for the first time that this thought-based control is actually possible. Okay, so I'll just briefly take you through the setup. So here, what you see is, here you have this monkey, and the monkey has to do a task. So the task is, you see this red cursor, that red cursor needs to be brought to this target, which is this round thing, and the monkey can use the joystick to bring the cursor to the target. That was the task. Okay? So that was the task, and while the monkey is doing the task, her neural activity was recorded. So electrodes were implanted in her brain, and then that signals from the electrodes were sent to the computer. Then the neural activity was encoded, it was decoded, and then the decoded output was sent to the robotic arm, which could then do some action. Okay? So the monkey did the job perfectly because they were trained to do this kind of tasks. But what happened later on, which is interesting, is after a while, you see the monkey is doing the same task but not using the joystick at all. So the monkey is controlling it just by thinking about it. So moving the cursor to the target just by thinking. So this is called motor imagery. So you're imagining that you're moving it and you can actually move it. Okay, so this was a sort of very exciting time for PCI community in general. Okay, and then we also have some a group in Austria, GRAS, that they did experiments with patients who are paralyzed. And let's see if this video works. It's working. So essentially, let me try to see whether I can play. Okay, so essentially what was happening is that this patient was given a EEG. EEG is a recording technique where you record the brain signals, and then what you do is essentially you try to record training. This person was trained for a month, okay, and then what he could do is he can actually control his motor action, something that he couldn't do on his own because he had spinal cord injury, but he was able to sort of regain certain function just by a month of training. Let me try to find this. One second. Yeah. So this is without any stimulation. He cannot move his finger. Okay? And then he is wearing a cap. Okay, so this is called functional electrical stimulation. So his brain signals are being decoded and fed to some circuit here, so that will enable him to move. So now you will see, yeah. So this is EEG cap. So there are electrodes inside the cap that records the brain signal. So some training was given to him so that he could control, his. He just cannot do it at one go with some sort of. So he imagined moving the foot. So you can imagine whatever that maps to the output. Doesn't really matter. It doesn't have to imagine holding the glass. And now after the training, now you see. So this is in real time. So his brain data is being fed to the circuit in his hand. So now you can see that he will actually grasp the flex his fingers as well. And extend the fingers, which we couldn't do before. At the moment the stimulation stops, the finger drops. Okay? So it's not that he had to have it all the time. Otherwise, the moment you stop giving the stimulation, then you're back to square one. So then there were a lot of these very games also, and then there are like small electronic electrodes you band like this. With that you can control, play ping pong, make a phone call, things like that. And then there were a lot of games that were developed also. But after that, that sort of hit a stagnant. But a couple of years back, the same lab that actually produced the first, monkey demonstration, they came up with this idea of brain-to-brain interface. So no more computer. Brain signal of one is being fed to another. So their model system was rats. So they had one rat. They recorded signal from one rat and fed that signal to another rat. So it was kind of the first rat was controlling the second rat. So whatever the first rat was doing, the second rat was doing the same thing. So normally what happens in these kind of tasks is you always have the animal perform the task many times before, so that the animal is familiar with the task. With this, what they did was the first animal, the first rat was familiar with the task, the second rat was not familiar with the task. But still, the signal from the first rat, when it was transmitted to the second rat actually performed the new task with the help of the signal from the first rat. Okay. And then, this is Professor Rajesh Rao, he's in University of Washington. He tried to do this. So his brain signals were passed on to another professor who was in another building. And they said that he could receive some, but we are more resilient, so the signal needs to be more strong. Okay. So there is one, hopefully this complete. Yeah. So there is a company called Backyard Brains. So if you're interested in, these are meant for high school. So they gave this, kits that you can do this kind of, just with the microprocessor Arduino kit, you can program and do those things. So here what they do is, just stick to the main part. Yeah. So this is a volunteer. So she's having her electrodes placed in the arm. Okay, and instead of brain, because it's difficult, so they are just recording it from from here. So it is getting the subject prepped. So you can just flex this muscle like that. So she is controlling her own, right? So this is fine. And then you can look at the signal. So yeah, so these are like, whenever you flex the muscle, you see this muscle noise. Okay. So you can see there's a lot of this muscle noise. Okay. Now, what is interesting is they have another volunteer. Okay. And now what is happening is her signal will be fed to the second volunteer. So the second volunteer is not going to be voluntarily flexing his arm. The first volunteer's arm signal is being passed on to this. Okay, the second guy. Okay. So the girl's signal are being passed on to the guy. Okay. These are the kits that they distribute. It's not very expensive. Yeah. So now her signal is being transmitted to the second volunteer. So now, so he is not voluntarily doing it. Just the signal is being passed on. So that makes him do it. So that's what it says, that her brain is controlling both the arms now. So that is something that you can do. Okay, so now moving on to a little bit more knowledgeable. So, how do we perceive things? So we see some things, how do we know what is it that we are seeing? Okay. So there are two things, sensation and perception. Okay. So as you can see, if you look at both the squares here, so one is darker than the other, right? The top square appears darker than the other. Now if I just remove the background, right, you can see actually the squares are of the same color, same intensity. Because of the background, it just looks that one is darker than the other. Okay? So there is this thing between sensation and perception. So sensation is what the stimulus, the information what we get. And perception is essentially how we make a decision based on the sensory information that is presented to us. So whenever there is a mismatch, then we have what is known as illusion. Okay? So here if you look at this diagram, you can see the fish and the background is black. And then slowly, somewhere here, you can see the black bird. Okay? Now the fish becomes the background becomes the bird. Alright, at no point of time can you see both the bird and the fish together. Even if you think you are seeing, you're not. You're just doing it very fast. So you can have you're shifting from one percept to another. Okay? So now look at this picture. So can you see like a young girl with a hat, right? That everyone can see? Okay, and how many can see an old woman? Okay. So here, for those who cannot, so if you look at the eyes, this is the eye, this is the mouth. Okay? So these kind of illusions, because it is clashing with our sensory information, that is why we have this kind of percepts. Okay? Now how do these things, this representation happen in our brain? Alright? So it is represented, so there are many, it's very complex, so there's low-level processing which happens right in our retina, and then it goes to our brain, cerebral cortex, somewhere here, visual cortex, that is the main center of processing visual information. But how this data is represented in our brain, so in the late 18th century, a group of German philosophers and psychologists came up with this idea of gestural psychology. So the idea is that, this is a very common phrase that you all have heard, must have heard, that the sum is more than its parts. So that is what actually they propose. It's not really more. It is often wrongly misinterpreted. It's actually sum is different from its parts. Alright? So they said that our brains follow certain laws, and using these laws we group things together. For example, we group things that are nearby each other. We group things that are similar to each other. We group things that goes up and down at the same time. We like to close, we like continuity, we like symmetry, and we like to close a figure, okay, even if there is no closure that exists. Alright. So they also talked a lot about what is the main figure and what is the background. So this is again a very famous picture where there is a white vase, this is called Rubin's vase, and there are two black faces. Right? So either you see the vase or you see the face. You cannot see both, okay, at the same time. So, and then they also talked about spatial relationship of objects. For example, the bottom figure looks very weird when placed like this, but the same figure, if you just place it a bit back, then it falls naturally. So these gestalt psychology talks about these kind of spatial relationships between objects as well. And a lot of their ideas have now been proven true, not all of them are true. Okay? And then this illusory contours. For example, the bottom line looks longer than the top line, but if you just take off the ends, you can see that both are the same. So this is called the Mueller-Lye illusion, it's a very famous illusion. And talking of closer, this is called the Tanisa triangle. Although there is no triangle, neither a white line nor a black line, but our brains complete that. So we can always see a black triangle as well as a white triangle. We can only see that because we are familiar with the shape triangle, otherwise we can't. Okay. Now one last one. So if you look at this, does it remind you of anything? Can you make sense of what is it that is being shown here? Does it look like just random broken pieces of glass or something like that? Now if I just sprinkle some ink on it, now can you see what is behind? The letter B, right? So this is the same picture, but because it is occluded now by the ink, our brain fills it up for us. Okay. So these are the sort of things that gestural psychology talks about. Now where does machine learning come? Okay, so machine learning can be used in various areas in cognitive neuroscience. One of the areas is to classify the mental state we are in terms of brain-computer interface. The other one is we can use machine learning techniques to find neural markers to identify, diagnose some disease like Alzheimer's or autism. Then we also can look at visual perception and try to understand the mechanism by using machine learning methods. We can also, of course, find neural network models of cognition. So I'm just going to show some very important papers here. So this is a paper in 2001 where they showed images of faces and non-face objects. So in our brain there's a very special place for face processing because that is something we grow up with. So there's a dedicated center that just processes face. Okay, and if you have impairment in the face processing stream, you may end up with a disorder which is called prosopagnosia. That means you can't identify faces, but you can identify everything else. Okay. So using multivariate machine learning techniques, they could find areas which respond only to faces. They can find areas which respond only to place. Okay. And then this is another review paper where they talk about how you can sort of do a brain-machine interface. So this is a futuristic idea where you have electrodes implanted in your brain and then wirelessly that signal is sent to a, the rudimentary computer here like a fanny pack, and then that computer decodes your brain signal and then the data is sent to your arm, like the way you saw in the video where you can then do certain tasks. Okay, so that is, but we are not there yet as you can see. Okay. Machine learning can also be used a lot in disease diagnosis. So this is a review paper where it's different kinds of pattern classifiers were used to predict Alzheimer's disease versus non-Alzheimer's patients' data. Okay. And then this is another paper where they used actually Mahalanobis distance-based measure to find the difference between same type of pictures and different type of pictures. Okay, so face images and non-face images. Okay. And finally, nothing is complete without deep learning, so I will go into one, again, very nice review paper. So you can use the convolutional neural networks. And there are three such applications that comes to mind. One is that you can look at any new image and try to say what image it is. Okay? So here they have shown using a three-level deep convolutional layer, and then you have a two-layer later on that. This is a cat. The other application is that you can compress this image of cat into a low-dimensional case, and then again reconstruct the image later on. And the third is that you can predict. So you have a series of letters, say WHA. What will be the fourth letter? So that will be, with high probability, it will be T. So you can train your network, your neural network to predict what will be the text that's coming afterwards. Okay, so any sort of machine learning signal classification framework using neural data would look like this. So first you acquire the signal, you get the data, and then you do some pre-processing on the data because you never work with raw signals, and then extract informative features from the data, and then decode the data, send it to the output. So the output might be a robotic arm, might be a computer, depends on your application. So for data acquisition, you can have invasive things like in monkey, you saw the electrodes were implanted in the brain, but invasive is typically involved with non-human models because no one will allow, just for science to have their brain poked and put electrodes inside. But sometimes if you're lucky, you can get invasive human recordings because oftentimes patients who suffer from epilepsy, they have electrodes implanted in their brain and they wait for seizures to happen. So I was lucky to get such a data. So oftentimes what happens is these patients anyways have electrodes implanted in their brain and they were just waiting for seizures to happen. Then you piggyback your experiments on them and say, since you already have electrodes, can you do some task for us? And then they see some pictures, we record the signals because the signals are already there. So that kind of data is very useful because it's very less noisy. And non-invasive methods, which is typically what is used for humans, you have EEG, fMRI and MEG. These are the three most important sort of tools that we use in neural signals. Okay, so in terms of each have their pros and cons, mainly the problem is some have good spatial resolution. With that, what I mean is we can pinpoint and say which area in the brain is producing the signal. Some have very good time resolution, where we can say that what is happening in millisecond level in your brain, but we don't have best of the both worlds yet. Okay, so EEG is something that is very good with time, but not so good with space. fMRI is something that is very good with spatial resolution, but not so much of time because there is a 5 to 7 seconds delay. Okay. So again, in our lab we do a lot of EEG experiments. It's one of the oldest methods, first developed in 1924. Essentially it measures the voltage difference, the potential difference between 2 points. We place electrodes and measure the potential difference. Okay, now after that what we do is we measure these components. These are called ERP components when we do a task. So for example, this here is something called the P300 speller. So if you're trying to spell a word, so again go back to Stephen Hawking and see how you used to spell each thing. So say you're going to spell a word, whenever you see a letter that you want, then the brain will give you a signal exactly 300 milliseconds after you see the word. Okay, and that is something that you can catch. So how does it look like? So the blue line is the target signal, so that actually looks like the word that you want to capture. Okay, so you just look at this. Okay, this is the brain signal, and then you see that almost around 300 milliseconds after you see the letter, you get this very strong positive peak, and you can decode the letter from there. Okay. So, for example, now in machine learning, what is it that we're trying to do? So the idea is, this is again a supervised learning technique. So what we do is we have this, say, EEG implanted, EEG cap in our brain, and then we are looking at pictures and then recording the signals. Okay. Now, then we try to reverse engineer that. So given any unknown signals, okay, the goal is to, based on that unknown signal, we want to predict whether it belongs to class A or it belongs to class B. Okay? So that is what we are trying to do in machine learning classification. So given any unknown data, we try to predict what is it. So it's a supervised problem. You can also have unsupervised problem. But the problem, what is the challenge, is that neural data is very high dimensional and extremely noisy. So to get informative features out of the neural data is very difficult. So for example, here you see this was an experiment where there are 81 electrodes and it is recorded at 1 second, 200 hertz. Right? So the total data dimension is over 18,000. Okay, and here are the two classes. So for example, here the target, the green target appears on the left of the screen, the red dot is the fixation, and the target appears on the right of the screen. This is one such image, you have many such data. Okay. As you can see, just by looking at it, it's impossible to differentiate between the two signals. And human brain is supposed to be the most advanced, computational machine that we know of, even we can't differentiate easily between the two things. So, because both are very similar, so it's very noisy. So it becomes problematic for us to just use standard machine learning techniques to neural data and come up with a good classification. So we have to sort of do something else. So the major challenge is that this high dimensional space is very challenging, even with state-of-the-art computing architecture. Okay, so there are a lot of computational issues. Just inverting a 100 by 100 matrix itself is difficult at times. The other thing is we often have what is known as the small sample size problem, because these are human data, and our data, as you saw, the last example was over 18,000 dimension. We cannot expect a human participant to do the experiment 18,000 times just because we want well-conditioned covariance matrix. That doesn't really happen. So oftentimes we have very few training data from which we have to sort of estimate the parameters. So the solution is that we extract from the data the informative features. So there are many algorithms. It's called feature extraction. You can also have feature selection. So the idea is you reduce the data dimension, but you also keep the informative features. So there are two ways you can go about it. You can do an ad hoc or heuristic approach, and that is what is mostly done, because if you have some prior information, for example, if you know that one electrode is what you want to look at, because that is the informative electrode, then you don't care about the electrodes from the other places, you just look at one electrode data. But oftentimes these heuristic approaches end up being suboptimal. The other issue is you can use an algorithm and let the data figure out what is important. Okay? So, in heuristic approach, oftentimes what we do is we rank the electrodes based on, say, some scores. But it could be suboptimal. And oftentimes what we do is these are all spatiotemporal signals. Right? So we can divide it. We can process the signals first in space and then in time. So we can, implicitly assume space-time separability. But that again is not ideal. You can also go to the frequency domain and look at the spectral features, but also the features could be suboptimal. Okay? So let me just give you an example. So in this 81 electrodes data, so this is the plot of the best two electrodes. So this is the left and this is the right. This is the best electrode. Okay, that gives, so the idea is we want these two distributions to be as separate as possible. Okay. And then this is the second best electrode. So you can see these, and this is again the best electrode and the 10th best electrode. So now if we want to concatenate both these electrodes together, okay, so this is how this is going to look. Okay. Here is the concatenation of the top two electrodes. Here is the concatenation of the first electrode and the 10th electrode. So ideally what we are looking at is the more overlap the data is, the bad it is. The more separated the data is, the good it is. Like that is what we are looking for. So if we actually calculate the error, we see that the error is more when we take the top two electrodes rather than taking the top electrode and the 10th electrode. And why does this happen? This happens because, for example, it might be that both the electrodes, the top two, are giving you redundant features. They are from the same place. Whereas the 10th electrode is from a different place in the brain. It is giving you new information. Okay? So oftentimes it's not a good idea to just, take the top features. So in algorithmic approach, the idea is you have some transformation, you have some mapping essentially, where you map your n-dimensional data space to n-dimensional feature space, where your n is much smaller than n. Okay. So principal component analysis is one of the most used, popular method for reducing the dimension. Then you have something called ICA, or independent component analysis, and you have linear discriminant analysis. These are just like very standard classical methods of reducing the data dimension. Okay. So I don't know how many of you are familiar with PCA. Anyone? Oh, okay. Good. So essentially, multidisciplinary component analysis is a simple second order best technique where you just you do an eigen decomposition essentially, and you try to find the projection. So you have some high dimensional data, you want to project it to a very low dimensional space. What you do is you just take the eigenvectors corresponding to the highest eigenvalues of the covariance matrix. That is what you do. Okay. So here it always finds an orthogonal basis for the given data set. So you think of it as rotating the data so that you find best representation of the data. Okay. Now linear discriminant analysis, again PCA, the problem is you don't give importance to class labels. So you treat the data as whole. You don't give importance to class A or class B like that. So LDA actually gives importance to class level as well. For example, if you see, these are two classes. Right? If I use PCA, what we're trying to do is say reduce this data to a 1D feature. We want to extract, reduce these 2 points, 2D points, and we want to reduce to 1D. Okay. So PCA would, the projection would be in a line like this. So if you project, you can see a lot of the points are overlapped. However, you can see that there exists lines where you can actually project it nicely, but the classes will be well separated. And that is what you ideally want. So, and that is the objective of LDA, is to project the data, reduce the dimension, but also keep the classes as separated as possible. Okay. So for classification, often LDA is preferred. So here, for example, let me try to give you very briefly what LDA is. So here, one possible function might be the difference between the means of the two classes. Okay, so if you just project in the direction of the difference of the means, then we might end up getting a good, separability. So here, what happens is even if you just project in the direction of the mean, you can see that there will still be overlap. So what we are missing is we are not taking into account the class scatter. So how scattered the data points are around the mean. So we can do that, we can have the class scatter and then normalize the difference of the mean by the class scatter. Okay, and then end up with a objective function like this, which is just the difference of the mean and then divided by the class scatter for each class. And that is nothing, but that is what LDA is all about. Right? So, but there are certain limitations in LDA because the feature space is dependent on the number of classes you have. So it always has to be 1 minus the number of class. Also, LDA fails if you have scenarios like this where the mean is overlapped. Then you don't get a good classification with LDA. Also, you can show theoretically that it is optimal when the class scatter is uniform. For example, when you have a, say, circle. So, or you have exactly the same covariance matrix for all the classes. Okay. Now, what we can do is we can think of a very simple method where we use PCA, but now not in the entire data, but using just one class per class. Okay. So the idea is to catch this underlying data manifold. So imagine this is a very high dimensional space. There are 3 classes A, B, C. Right? Red, green and blue. Now the objective is we want to catch this data manifold. So if we just use normal PCA, you can see that it will be overlapped. We can use independent component analysis. In this case, it will give us good separation. So independent component analysis essentially projects the data in the direction where it is least Gaussian. Okay? But unfortunately, we cannot use ICA for low sample size problem. We first have to use PCA, reduce the dimension, and then use ICA. So the moment we use PCA, that sort of the overlapping happens. So instead, what we can use is, we can just use PCA for each class. So PCA in class 1, PCA in class 2, PCA in class 3. That way, we can catch the data manifold nicely. Okay? And that is exactly what we do. This is one method. So it is called class-wise PCA. It works very well with high-dimensional data because it's PCA. You can just reduce the dimension, and because we are applying it for each class, we also give importance to the class separability, which was not there in the actual PCA. And it is non-linear in the sense, non-linear, this is a very like engineering computer science way of saying non-linear. That just means that we don't have one transformation matrix. We have as many transformation matrix as the number of classes. So, what are the steps? We automatically discard the noise, extract the features, and then design the classification based on which we can say that which class an unknown data belongs to. Ultimately, we want to classify. So, an unknown test data comes, we have to say whether it belongs to class A or class B. That is the whole goal. Okay. So, if we take an example here, so there are two classes, so omega 1 and omega 2. So, this is the training data, this is the mean. Okay. For in the step 1, what we do is, so X star is our unknown test data that we want to classify whether it belongs to class omega 1 or class omega 2. Okay. So what we do is first we project all the data, the training data, the test data, everything onto the first PCA subspace S1. Okay. Once we do that, then what we can do is we can calculate the posterior probability using the base rule. Okay. And then whichever class has the highest posterior probability, we can say that this data belongs to this class in that subspace S1. Okay. Next, we do the same thing, but for PCA, the second subspace. Okay. Again, we project the data and the training data, and then find the class membership in the second PCA subspace. Finally, what we end up is that we reduce the data in these two PCA subspace. We have two possible labels. One label in PCA subspace might be omega 1, in PCA subspace 2 might be omega 2. But ultimately, you have to come up with one label. So, how do you do it? What you do is, because it is posterior probability, what we can do is we can now compare between the two posterior probabilities and then assign the class that has the highest posterior probability. Okay. And that is what this method is. A very simple method. It is second order method, is very quick, real time. And that actually works. Okay, so this method was used, it's not made for some reason. Anyways, so ideally this is a patient that was, he is completely paraplegic, so he couldn't move. So you can see the harness is fitted for him to stand. And then what happened was real time, if the video was played, see, he started walking with a walker, but he could take the steps. And real time what is happening is we are using the same method that I just described, decoding the brain signals and then passing it on to the functional stimulations in his leg, and that enables him to walk. But he is using a walker, but he could take the steps. Okay, and this happens because the method we are using is very fast. You can decode it in real time. Alright. So if you aim for a very complicated method, it might not work in applications where you really need fast encoding and decoding. Okay. We also did, we also dabble in other types of, method like mental health disorders. Here we used features which were used topology. So we actually do something called topological discriminant analysis, where we use persistent homology. So the idea is we are trying to get features that are persistent. So essentially we're trying to look at the peaks and valleys and we use filters in visual complex, get persistent features. We do this persistent diagrams. From that we get something called persistent entropy. Once we get this TDA features, then we use any machine learning techniques that you want. This is actually linear SVM. And then you can see that this is an autistic data where we show that using this topological features we get a much better classification rather than just normal classification. Alright, so some of the other things that we do in our lab is trying to develop algorithms for this kind of very noisy neural data. So we currently are using functional data analysis. So the idea is we convert the data into functions. So that way the dimension problem goes because we only work with the functional coefficients. and then work on the functional space and try to classify and do everything on the functional space. And then the other thing what we do is we do a lot of studies on face perception, how do we understand face, visual search, which is how do we look for some objects in a very cluttered scene, and how do we find, how do we know from our brain signals which part of the image we are concentrating on or we are attending to. And then recently we have been starting to work on neuroeconomics, right? So trying to see how we are emotions play a role in our social decisions. So we are using an ultimatum game, and then we are showing pictures of emotional stimuli, and then seeing how benevolent are we when we look at someone who's happy versus someone who's sad or someone who's disgusted, things like that. And then we also are doing some experiments with Indian food and trying to see what kind of craving, what is the neural mechanism behind food craving, because obesity is India is a country that is double, and we also have very huge number of malnutrition kids. We also have currently lot of obesity. It's mainly in the urban cities, but obesity is a growing problem in India, and the rate is greater than the global average. So obesity is going to be a big problem, and nothing much has been done in Indian food at all. And we have just started. We just had one work, and we're currently writing up the other one. But we're trying to see whether we can find in Indian population some sort of markers of how we can control our craving, or what using some intervention whether can we control our craving for not so healthy snacks and increase our craving for healthy snacks. So that was something that we are currently working on. And I think all of you must be hungry, so I'll just stop here and any questions.
So any questions or comments? Anybody, any questions?
This neural data when it's seen and when you make work in say some supplement you were showing some video where analyze what you were speaking of a glass, right?
Right.
So you were thinking something.
Yeah, so at that
then it was transmitted to that
right.
and that made it work.
Right.
So what goes in between receiving and then processing the data and then analyzing the data
and then so that is why you need, like, for that kind of real-time applications, you really need very fast methods. You cannot have like your deep learning and your neural networks, because that will take for ages. You have to, that is why you need, say, PCALD, like this very, like second order best methods. So these were just very simple methods, the ones with
classify first.
Right, you classify first. First you decode. First you encode. Encode, first try to see, find the, from the training data you find some baseline, find the parameters for the model essentially. And then once you have the test data, so once the new thing, so when, for example, the person is the video will play was walking, so that is the new test data. So you immediately classify and you know that whether you want to go left or you want to go right. And that is how it is fed to the output. So the functional electric stimulation. For example, if you are holding the glass, so that he was actually not thinking about holding the glass, because his brain worked better when he was imagining moving his feet. So he was actually imagining foot movement, but that was transformed. Doesn't matter, right? We just want to decode class A versus class B. Right? Or class A versus class B versus class C, doesn't matter. And then that output was fed to the decoded output, fed to the functional simulation.
And one more thing, you showed that data manifold.
Right.
And there you showed that if you do separately PCA, so PCAs look like the tangent lines to the data manipulation.
PCA, essentially what it does is it just projects it in the direction of the maximum variance.
But in the picture you showed, the PCAs look like white-wise tangents. In your picture there are three.
Right.
But they were looking like the tangent lines to the curve. Does that have some connection?
No, so essentially it's an orthogonal projection of the data onto a plane.
In that picture it looked like...
Yeah, the picture it looked like tangent, but it's not. It's essentially the eigenvector corresponding to the highest eigenvalue of the covariance matrix.
Okay.
Yes.
What I asked you, ma'am, might take you 12 years back in time to your Ramalinga Swami Fellowship days. I am from a humanities background, let me clarify, because I'm a non-mathematical person here. But how you fell under my radar is that I have been a sole caregiver to my mother who had vascular dementia. And all the literature that the Alzheimer's Association used to provide us, what she used to do is she would ignore the literature, go straight away to the footnotes and the bibliography, and then read the articles that she finds there. And this is how we discovered the research of Miguel Eckstein. And that is how you came in. And when we read your articles regarding, and what intrigued her was, it was one of the last things she read before she moved on, passed away. The use of the multivariate pattern classification analysis in the multivoxel patterns, and how a human factor that you mentioned there, that the emphasis was given rather on the individual's response, like those Lumina response pads that are made to wear, the centrist ones, where you have those 8 buttons, and then the responses that you give, and you gather the data, where the emphasis is given rather on the response of the individual rather than the stimulus conditions. So if it were the other way around, if you were to emphasize equally on the stimulus conditions rather than the individual's response, how would the data then change? I'm just curious about that.
So are you saying, let me understand this question. So something that we normally we do this experiments with many subjects, right? So and there's something called individual difference. So each subject there's a quite a bit of variability. So we sometimes if the variability is too much, then you have a problem. But ideally, you would like to have a sample size that's big enough so that you have different sort of clusters of behavior if there is any. But ultimately, if you're doing something, a pattern comes. So that pattern would be common to different individuals as well. So that is how typically, if you have a large enough sample, that is how it happens. If there is a pattern that is common to all and that is important for doing the certain task, that will show up. But if you take very few samples, then you might not get it. But if you have a good enough sample size, that stimulus condition, what is driving that stimulus condition will show up.
Here, I believe there were just a dozen student volunteers from University of San
Yeah, so normally, like if before we have student volunteers, typically it is student volunteers because we try to do basic mechanism. But if we are going for some, say, disease, then we need a patient population. So we're trying to do. We have some collaborations with AIMS, which has come up. So in the future, we would like to do some experiments on patients as well.
As we are running out of time, we will take last few questions. So, anybody?
Hello, ma'am. Ma'am, I have 2 questions. One is, how do the symptomal complexes exactly help in data analysis?
Oh, okay. So what we do is we use this filtered simplicial complex and get this persistent diagram. So essentially, from the persistent diagram, we get this birth and death rate. So how long a feature stays stable. So that we then sort of, because we need to vectorize it for ultimately to input it to a machine learning algorithm. So that length of birth rate minus death rate, that is what is each feature, means is integrated for each level, and then that gives us the persistent entropy. So persistent entropy is like a set of vectors that we just feed to that is the input to the parameters.
Another question is about that BCA. What about the ethical dilemma or say about cancelling the thoughts? I mean, if anyone has taken the thought, I think it will be an immediate action.
Yeah, there's a lot of like, you are talking about the brain-to-brain interface. Yes, there are like a lot of ethics, so I'm not someone very comfortable with the entire issue. So that was just sort of to say that if someone's brain is not working, the idea is then, but you can feed signal from someone's other brain to sort of complement your capability. But then there is an issue of free will and how much you are resisting and how much it, I mean, for the video that you saw flexing the hand, that is simple because that is not really brain signals. You are just having some stimulation here with automatic reflex action. But how much you are actually controlling the brain, that is a difficult and ethical question. That's why you don't see a lot of these research going on. It's still sort of very meaningful. Yeah.
Thank you, ma'am.
And on the last slide, you told that you can reconstruct the curves using the data points in the context of neural network.
Oh, neural network, yeah. So you can reconstruct. So essentially, you have an image. So that image pixels you can compress and save it in some convolutional layer. And then you can expand that convolutional layer and then get back, reconstruct that original.
Well, so it means since you are working in the context of neural network models, that means you are assuming some kind of activation function, right?
Yes, you have to assume
So
some
my
kind of.
concern is that I can take a lot of activation functions to construct the function. So, I mean, I can take any kind of functions.
Right.
So how that is
Normally what happens is we typically take a very simple activation function like the signal. Something like logistic. That is what is typically used. But I mean, there are now words actually. So normally neural network methods, you have a feed forward, you go, and then there's a feedback to correct your whatever the loss or the gain, and then again you do this, go this pattern. So nowadays there are like, I saw some papers that you have one go feed forward, and then you do a sort of Bayesian kind of analysis there. And don't do the feedback. Just do a Bayesian, try to estimate the parameters and then.
I mean, similarly, we can consider the gain function as well. I mean, is there any mechanism through which you can discard other activation functions?
I am not very sure whether like normally you don't see, I haven't seen any papers on comparative study of different activation functions and the result on neural network. But again, I don't really work in deep learning per se, so maybe there is, I'm not very sure. I just know of the papers that are very popular, that's what I read. But I don't think activation function, there are, normally what I see in most of the papers are there are a set of activation functions that are standard and that is what we use.
Thank you.
Yes, ma'am. So actually, once in your lecture, you said that the human brain is very complex, one of the most complex computational machines.
Yeah.
So my question is that is it really only a computational machine, or is there something else? Because if it is, then it might actually be impossible for us to prove that it is from Gödel's theorem. So, like, is there something else, or is it?
I mean, there are like a lot of this consciousness research, right? So then and there's also something called precognition or pre-consciousness. Truth be told, so I'm not too much into consciousness research because there are a lot of sort of interesting studies, but there's a lot of also noise. It's very difficult to distinguish the consciousness research, the noise from the signal. But yeah, maybe there's something, but whatever we cannot prove it or cannot see, it's hard to sort of make your hypothesis based on that, right? So some of the consciousness research are very solid. but not all. So lot of the times you see a lot of very like tall claims of consciousness, but ultimately if you go down and look at the research, they're like just being very simple. There are always some assumptions. So that way, maybe it's not just, yeah, so the group who did this brain-to-brain interface, they are of the belief that brain is just a simulation box.
Right.
So you can just put any signal and that will work. But then there are others who don't fall in that category.
Thank you.
Unfortunately, due to running out of time, we cannot have any more questions in this session. However, if you have any more questions, you may ask the speaker personally. Thank you. Thank you so much, ma'am, for such an interesting and interactive session. We would like to felicitate our esteemed speaker, Professor Koel Dash, and we would like to call Dr. Shubhabrato Dash to give her the gift of our gratitude.